It's tough to follow Jerry, who is one of my mentors, and we were sad to see him leave. There is a study of osimertinib, that adjuvant open, so we look forward to that. I'm excited to be able to give a talk where there's no data. Um, so if you want to snooze a little at the beginning, and then I'll maybe excite you about some opportunities here. Those of you like Dr. Crawford with his newfound love of finances might want to ignore it because they'll get angry. So first I'll talk about what data we do have with various vaccine therapies, both generic tumor antigens, tumor-specific antigens that have been tested, and then thoughts about designing better vaccines, but there are experts here who will certainly address that more thoroughly. Talk about adjuvant studies that are ongoing and upcoming, as well as my thoughts and my agreement with other people in the room about the importance of neoadjuvant investigation. First, with vaccine therapy, the advantages are very clear: low cost, low toxicity, although some of our newer vaccines may be more expensive to design, patient and tumor-specific potential for therapy. The disadvantages are clearly the time to onset of action, where early stage may be the ideal setting, and appropriate design of adjuvants has been lacking throughout historic drug development, but we are hopeful for the future. And then the real world experience has been just frankly bad drugs, bad trials, all negative. So what trials do we have in lung cancer now? I lumped the large trials together here, both metastatic disease and early stage, and the Magritte MAGE-A3 study was closed. We have consolidation studies. I do consider stage 3 early stage even after chemoradiation, so lump that into this talk. I'll show you the data for that. The START MUC1 vaccine study, everyone was very excited after the overall study population was negative. Now this is a study followed by after concurrent chemoradiation with stable disease, patients received either cyclophosphamide followed by tazemetostat, the MUC1 vaccine, or saline followed by placebo. And while the overall study was negative, there was an overall survival advantage in the patients who received concurrent chemoradiation versus those who received sequential, which then launched a very large, mostly Asia predominant study, I believe, of confirming this in patients who received concurrent chemoradiation, and that study was just stopped by Merck. It was considered non-promising. So those have failed. What about what we've seen with patient-specific vaccines? And Jerry started, and I completed a study of the adjuvant GVAX KRAS vaccine in patients with resected KRAS mutant lung cancer. It was only a feasibility study, and while it did elicit an immune response, it's unclear if there would be clinical significance or how to move that forward. And it probably isn't the most potent of vaccines. There are better designs out there now or in development. When preparing for this talk, I did a clinicaltrials.gov study, and there's some cool stuff going on out there, and these may be some of those better vaccines, better vectors. There's a mature dendritic cell vaccine, personalized at UWash, an autologous tumor transfection of fibroblasts at Pitt. And we have many tumor-derived vaccine startups out there that are looking to really find ways to personalize immunotherapy. So in designing better vaccines, I think we need better and more immunogenic vectors, appropriate antigen selection. The antigen doesn't only have to be expressed in the tumor, it has to be immunogenic and potentially requiring some sort of patient HLA selection. And then better design of adjuvants, and perhaps that's where our checkpoint inhibitors come into play, not as a typical adjuvant, but as a potential adjuvant. So now let's talk about adjuvant checkpoint inhibitors, and this is where we have absolutely no data, but there's been a lot of excitement and there's a lot ongoing or upcoming. So I hope these project and you can see them. This is the PEARL study. This is Merck's study. It's being run in the EURTC. It's a very large study of, I'm basically going to walk you now through the differences between the drugs being tested and their schemas. It's a study with a typical adjuvant patient population, R0 resection, 1B greater than 4 centimeters through 3A, up to 4 cycles of chemotherapy, pembrolizumab for a year or placebo, and the primary endpoint is disease-free survival. Now this is a huge study, and I didn't have access to the statistical plan. It's 1,380 patients. But presumably with that sample size, they're going to be able to report on a positive outcome with a very small benefit. So while I differ from some of the opinions expressed already and do think that recurrence-free or disease-free survival is exceptionally important, and it led to the approval of ipilimumab in resected melanoma, this is a disease-free survival study that's so large that even if it's positive, will it be clinically significant? And one could argue no, I don't know yet. We will see. So this is the adjuvant atezolizumab study, or MPDL328OA. Virtually the same schema, except for this study is requiring cisplatin-based chemotherapy and only selecting for patients who are PD-L1 positive. I applaud them for using a biomarker to select, although it might not be the best biomarker, and you'll hear more about that later. It's an interesting study design, or same study design basically, but randomized to best supportive care. And the primary endpoint is disease-free survival. Now, what problems do I see here? Without a placebo, disease-free survival is a little troublesome. There's a difference in when you're going to see these patients, how much confidence you have about what's happening to them. You're like, basically your bias, our bias as to when they might be scanned. So this is also a large study, 145 patients, and it's launching. I don't know that it has any sites off the ground and running yet. I'm biased, full disclosure, I wrote this study, so I'll tell you what it is, you can judge it. This is the adjuvant nivolumab via the NCI, and it will become the third arm of Alchemist. It's a study of the same patient characteristics, again, EGFR and ALK wild type. And while we do expect that most patients will receive chemotherapy, they're not mandated to receive chemotherapy, and postoperative radiation is permitted. It's a randomized study of nivolumab versus best supportive care with an overall survival primary endpoint. It is a co-primary with disease-free survival, so we will get a readout of this study early, but overall survival follow-up will be mandated. Now, we really hope that of those 500 patients who screened for Alchemist and the probably 475 who didn't have a mutation, those are perfect targets for this study. And this was approved with modifications by the central IRB this week. It should be open in the next many months. So neoadjuvant immunotherapy, could it be better? And I would say hands down the answer is yes. And we need to convince industry of this to commit to it. So these are the upcoming and ongoing efforts that I know of. I'm sure there are many, many others. That's what the MSLs tell me. But clinicaltrials.gov hasn't gotten them on their radar yet. So there's a Stand Up to Cancer Fund in neoadjuvant nivolumab for resectable non-small cell lung cancer between Johns Hopkins and Memorial Sloan Kettering. Only 2 patients have been treated despite being open for a while. It's been hard to get up and off the ground, but we do anticipate there will be some very important clinical data, correlative data that will derive from this study. There is a neoadjuvant durvalumab and tremelimumab study for resectable 3A and 2 disease. I know that resecting 3A and 2 is a very memorial-centric thing, but we're going to try to give them immunotherapy first. And then the LCMC effort that was mentioned yesterday, and the SEMA presented, so I won't show it for redundancy. But again, looking at neoadjuvant atezolizumab. So what's the argument for induction? This is easy with immunotherapy. Your tumor is in vivo. So is that necessary? We don't know if the micrometastatic disease has with it sufficient microenvironment to have benefit from these checkpoint inhibitors, but time will tell. Those studies are already launched. We can monitor for response. That's equally as important. Why continue with therapy if it's not working? And we heard yesterday there are financial models to contribute to that thought. Selective use of adjuvant cytotoxic chemotherapy. So if you are in that 20%, we know, unlike the TKIs, there are these very, very long-term responders who may be cured. So could we not administer adjuvant cytotoxic chemotherapy to the patients who benefit? And then I think it's a great platform to evaluate either chemo-IO combos or sequencing. And Dr. Carbone might kill me, but I keep pushing the issue. I think that in the LCMC neoadjuvant atezolizumab study, the patients who do not respond, which is going to be probably 80%, could be sequenced to chemotherapy. And we could learn a lot because we all have those patients in our clinics who fail these drugs and then have an absolutely remarkable response to chemo. So I think I've already brought up my future directions. Biomarker selection for adjuvant therapy would be important. Adjuvant use of combination therapy for high-risk patients. How do we define high risk is a question of debate. Induction therapy with sequencing and combos could be really, really useful. And then in adjuvant vaccines and checkpoint inhibitors, I'm sure these studies are coming, and whether they're personalized or not, we'll see. Thank you.
Thank you, Jamie. So any questions? I think Accrual and Alchemist will greatly benefit just as a comment overall, right, by having that third arm, so that everybody will have something that can be offered out of that protocol. So I think it's going to be a great addition.
And I think
John?
there will be a lot of disappointment across the country that the patients with EGFR mutations can't receive adjuvant immunotherapy. But I would say that to date, our data would suggest that it's the right thing to do. We have something else for them, and I don't know that they benefit as well from these therapies.
So I agree with you completely that the neoadjuvant approach, especially for testing, because if you start looking at the number of potential things we want to look at from immunotherapy, it's mind-boggling, and you can't do 800 and 1,300 patient trials. My question to you, though, is we know in breast cancer with chemotherapy and potentially anti-HER2 directed therapy that pathological CR is a marker for potential long-term survival in this surrogate endpoint. With immunotherapy and in lung cancer, do we know that, and how can we get to a comfort level that we have a marker in the neoadjuvant setting that we can say, okay, this is the right combination? Because we probably have more drugs and ideas than we know that, in the neoadjuvant, this is what I'm going to do. So what's your thoughts on?
So the pathology group at MD Anderson has done a large study of their neoadjuvant chemo patients, looking at sort of our version of pathologic complete response, because it just doesn't exist in lung cancer. And they looked at a threshold of 90% tumor response, so 10% tumor viability or less, and saw a fantastic correlation with survival. There just aren't enough studies. I mean, the breast data to support a surrogate endpoint of path CR is based on tens of thousands of patients, and we just don't have that in lung cancer. My colleague Matt Hellman wrote a paper in the Lancet Oncology looking at this it's a review and opinion piece looking or supporting major pathologic response, which is this 10% threshold. Now, how will that pan out in the immunotherapy setting? I don't know. I think the two small studies that we have or are participating in will give insight into that. They're both going to evaluate pathologic response, but we will need and the atezolizumab study certainly is large enough where evaluation of pathologic response will be informative. But even looking back through our chemo studies, they're not large enough to have to truly meet the definition of a surrogate or have anyone believe it. Yet they're worth looking at.
Thank you very much.